Hey guys, welcome back to another episode of my channel. My name is Keika. We're going to be going over concept 5.5. Nucleic acids store, transmit, and express hereditary information. If the primary structure of polypeptides determines a protein shape, what determines primary structure? The amino acid sequence of a polypeptide is programmed by a discrete unit of inheritance known as a gene. Genes consist of DNA, which belong to the class of compounds called nucleic acids. Nucleic acids are polymers made of monomers called nucleotides. Okay? And just to click on nucleic acid, a polymer, polynucleotide consisting of many nucleotide polymers. Nucleic acids, a polymer, polynucleotide consisting of many nucleotide monomers, serves as a blueprint for proteins and through the actions of proteins for all cellular activities. The two types are DNA and RNA. Okay? So that's nucleic acids. So I wrote for my notes here, the amino acid sequence of a polypeptide is programmed by a discrete unit of inheritance known as a gene. Genes consist of DNA, which belongs to the class of compounds called nucleic acids. Nucleic acids are polymers made of monomers called nucleotides. Okay. The role of nucleic acids. The two types of nucleic acids, deoxyribonucleic acid and ribonucleic acid, DNA and RNA, enable living organisms to reproduce their complex components from one generation to the next. Unique among molecules, DNA provides directions for its own replication. DNA also directs RNA synthesis and through RNA, controls protein synthesis. This entire process is called gene expression. So here we see a figure 522, from DNA to RNA to protein. Okay, so number 1, synthesis of mRNA in the nucleus. DNA synthesizing mRNA, synthesis of mRNA in the nucleus, and then from here it moves through what seems to be a protein in the membrane, leaves the nucleus, and the movement of the mRNA into cytoplasm via nuclear pore. Oh, it's a pore, not a protein, but a pore. So just a hole. Okay, into the cytoplasm, which is this yellow area here, and synthesis of protein using information carried on by mRNA. And so the mRNA goes to a ribosome, and once it's at the ribosome, then it synthesizes a protein. Okay? DNA is the genetic material that organisms inherit from their parents. Each chromosome contains one long DNA molecule, usually carrying several hundred or more genes. When a cell reproduces itself by dividing, its DNA molecules are copied and passed along from one generation of cells to the next. The information that programs all the cell's activities is encoded in the structure of the DNA. The DNA, however, is not directly involved in running the operations of the cell, any more than computer software by itself can read the barcode on a box of cereal. Just as a scanner is needed to read a barcode, proteins are required to implement genetic programs. The molecular hardware of the cell, the tools that carry out biological functions, consist mostly of proteins. For example, the oxygen carrier in red blood cells is the protein hemoglobin that you saw earlier in figure 518, not the DNA that specifies its structure. How does RNA, the other type of nucleic acid, fit into gene expression, the flow of genetic information from DNA to proteins? A given gene along a DNA molecule can direct synthesis of a type of RNA called messenger RNA, or mRNA. The mRNA molecule interacts with the cell's protein synthesizing machinery, the ribosome, to direct production of a polypeptide, which follows into all or part of a protein. We can summarize the flow of genetic information as DNA to RNA to protein. The sites of protein synthesis are cellular structures called ribosomes. In a eukaryotic cell, ribosomes are in the cytoplasm, the region between the nucleus and the cell's outer boundary, the plasma membrane. But DNA resides in the nucleus. Messenger RNA conveys genetic instructions for building proteins from the nucleus to the cytoplasm. Prokaryotic cells lack a nuclei, but still use mRNA to convey a message from the DNA to ribosomes and other cellular equipment that translate the coded information into amino acid sequences. Later, you'll read about other functions of some recently discovered RNA molecules. The stretches of DNA that direct synthesis of these RNAs are also considered genes. Okay. So for my notes, I wrote, DNA and RNA are nucleic acids that enable living things to reproduce their complex components from one generation to the next. DNA does its own replication and RNA synthesis, thus protein synthesis. Gene expression. DNA to RNA to protein. Each chromosome contains one long DNA molecule that carries several hundred or more genes. When cells divide, DNA molecules are copied and passed along from one generation to another. Codes for cell activities are within the structure of the DNA, and proteins implement the genetic programs. Okay? A gene in a DNA molecule can direct the synthesis of a type of RNA called messenger RNA, or mRNA. These are important. I'm going to bold that. mRNA molecules interact with the cell's organelles, ribosome, with ribosomes to direct production of a polypeptide, which folds into either all or part of a protein. Protein synthesis occurs in the ribosomes. In eukaryotic cells, ribosomes are in the cytoplasm while DNA is in the nucleus. mRNA brings genetic information for building proteins from the nucleus to the cytoplasm. This is important. From the nucleus to the cytoplasm. In prokaryotic cells, there is no nucleus, but they still use mRNA. Okay. So, the components of nucleic acids. Nucleic acids are macromolecules that exist as polymers called polynucleotides. As indicated by the name, each polynucleotide consists of monomers called nucleotides. A nucleotide, in general, is composed of 3 parts: a 5-carbon sugar, a pentose, a nitrogen-containing base, nitrogenous, and 1 to 3 phosphate groups. The beginning monomer used to build a polynucleotide has 3 phosphate groups, but 2 are lost during the polymerization process. Polymerization process. The portion of a nucleotide without any phosphate group is called a nucleoside. Okay. So here we see in this chart, this figure, we see pyrimidines and purines are nitrogenous bases. We also see sugars. Okay. To understand the structure of a single nucleotide, let's first consider the nitrogenous bases. Each nitrogenous base has 1 or 2 rings that include nitrogen atoms. They are called nitrogenous bases because the nitrogen atoms tend to take up hydrogen ions from the solution, thus acting as bases. There are 2 families of nitrogenous bases, pyrimidines and purines. A pyrimidine has 1 6-membered ring of carbon and nitrogen atoms. The members of the pyrimidine family are cytosine, thymine, and uracil. Purines are larger, with a 6-membered ring fused to a 5-membered ring. The purines are adenine and guanine. The specific pyrimidines and purines differ in the chemical groups attached to the rings. Adenine, guanine, and cytosine are found in both DNA and RNA. Thymine is found only in DNA, and uracil only in RNA. Now let's add the sugar to which the nitrogenous base is attached. In DNA, the sugar is deoxyribose. In RNA, it is ribose. The only difference between these 2 sugars is that deoxyribose lacks an oxygen atom in the 2nd carbon in the ring, hence the name deoxyribose. So far, we have built a nucleoside, base plus sugar. To complete the construction of a nucleotide, we attach 1 to 3 phosphate groups to the 5 carbon of the sugar. The carbon's numbers in the sugar include an apostrophe, the prime symbol. With 1 phosphate, this is a nucleoside monophosphate, more often called a nucleotide. Okay, so for my notes, I wrote nucleic acids are macromolecules that exist as polymers called polynucleotides. Monomers equal nucleotides. A nucleotide contains 3 parts: 5-carbon sugar, pentose, nitrogen-containing nitrogenous base, and 1 to 3 phosphate groups. The beginning monomer that starts a polynucleotide has 3 phosphate groups, but loses 2 during the polymerization process. The parts of a nucleotide that don't have a phosphate group is called a nucleoside. The 2 families of nitrogenous bases. Pyrimidines. And purines. Pyrimidines has one. Pyrimidine has 1 6-membered ring of carbon and nitrogen atoms, and consists of cytosine, thymine, and uracil. Purines are larger, with a 6-membered ring fused to a 5-membered ring, and consists of adenine and guanine. So this is important to know the difference between the 2. Pyrimidine has 3 types, purines have 2 types. Adenine, guanine, and cytosine are found in both DNA and RNA. Thymine is only in DNA, and uracil is only in RNA. This is also important. Please, take note of this. This one is, you need to know this. Sugar in DNA is deoxyribose, and sugar in RNA is ribose. Deoxyribose lacks an oxygen atom in the second carbon in the ring. And lastly, 1 to 3 phosphate groups to the 5 carbon of the sugar. Results in nucleotide. Okay? Yes. Nucleotide polymers. The linkage of nucleotides into a polynucleotide involves a condensation reaction, which you will learn the details in Concept 16.2. In the polynucleotide, adjacent nucleotides are joined by a phosphodiester linkage, which consists of a phosphate group that covalently links the sugars of 2 nucleotides. This bonding results in a repeating pattern of sugar phosphate units called sugar phosphate backbone. Note that the nitrogenous bases are not part of the backbone. The 2 free ends of the polymer are distinctly different from each other. One end has a phosphate attached to a 5 carbon, the other end has a hydroxyl group on a 3 carbon ring. We refer to these as the 5' and the 3' respectively. We can say that a polynucleotide has a built-in directionality along its sugar phosphate backbone, from 5 to 3, somewhat like a one-way street. The bases are attached along the sugar phosphate backbone. The sequence of bases along a DNA or mRNA polymer is unique for each gene and provides very specific information to the cell. Because genes are hundreds to thousands of nucleotides long, the number of possible base sequences is effectively limitless. The information carried by the gene is encoded in its specific sequence of the 4 DNA bases. For example, the sequence 5 AGGTAACTT3 means one thing, whereas the sequence 5 CGCTTTAAC3 has a different meaning. Entire genes, of course, are much longer. The linear order of bases in this gene specifies the amino acid sequence, the primary structure of a protein, which in turn specifies that protein's 3D structure and thus enabling its function in the cell. So nucleotide polymers. The linkage of nucleotides into a polynucleotide involves a condensation reaction. Adjacent nucleotides are joined by phosphodiester linkages, which consists of a phosphate group that covalently links the sugars of 2 nucleotides. The repeating pattern of sugar phosphate units called sugar phosphate backbone. One end of the polymer has a phosphate attached to a 5 carbon. The other end has a hydroxyl group on the 3 carbon. 5 end to 3 end, one end street. Bases are attached along the sugar phosphate backbone. The sequences of bases along a DNA or mRNA polymer is unique for each gene and provides very specific information to the cell. The linear order of bases in a gene specifies the amino acid sequence, which in turn specifies that protein's 3D structure, and thus enabling its function in the cell. The structure of DNA and RNA molecules. A DNA molecule has two polynucleotides, or strands, that wind around an imaginary axis, forming a double helix. The two sugar phosphate backbones run in the opposite 5-3 directions from each other. This arrangement is referred to as antiparallel, somewhat like a divided highway. The sugar phosphate backbones are on the outside of the helix, and the nitrogenous bases are paired on the interior of the helix. The two strands are held together by hydrogen bonds between the paired bases. Most DNA molecules are very long, with thousands or even millions of base pairs. The one long DNA double helix in a eukaryotic chromosome includes many genes, each one a particular segment of the double-stranded molecule. So here we see DNA. We see the double helix. So this is 5-3 here, and then 5-3 the other direction. And we see the sugar phosphate backbones in blue, and we see the hydrogen bonds between the bases on the inside, which are the bases are the yellow, and they're connected by hydrogen bonds. Okay, base pair joined by hydrogen bonding. In base pairing, only certain bases in the double helix are comparable with each other. Adenine in one strand always pairs with thymine in the other, and guanine always pairs with cytosine. Reading the sequences of bases along one strand of the double helix would tell us the sequence of bases along the other strand. If a stretch of one strand has the base sequence 5AGGTCCG3, then the base pairing rules tell us that the same stretch on the other strand must have the sequence 3TCCAGGC5. The two strands of the double helix are complementary, each the predictable counterpart of the other. It is this feature of DNA that makes it possible to generate two identical copies of each DNA molecule in a cell that is preparing to divide. When the cell divides, the copies are distinguished into daughter cells, making them genetically identical to the parent cell. Thus, the structure of DNA accounts for its function in transmitting genetic information whenever a cell reproduces. RNA molecules, by contrast, exist as single strands. Complementary base pairing can occur, however, between regions of two RNA molecules or even between two stretches of nucleotides in the same RNA molecule. In fact, base pairing within an RNA molecule allows it to take on the particular three-dimensional shape necessary for its function. Consider, for example, the type of RNA called transfer RNA, tRNA, which brings amino acids to the ribosome during the synthesis of a polypeptide. A tRNA molecule is about 80 nucleotides in length. Its functional shape results from the base pairing between nucleotides, where a complementary stretch of the molecule can run anti-parallel to each other. Its functional shapes results from base pairing between nucleotides, where complementary stretches of the molecule can run anti-parallel to each other. Note that in RNA, adenine pairs with uracil, thymine is not present in RNA. Another difference between RNA and DNA is that DNA almost always exists as a double helix, whereas RNA molecules are more variable in shape. RNAs are versatile molecules, and many biologists believe RNA may have preceded DNA as a carrier of genetic information in its early forms of life. Okay. So my notes. A DNA molecule has two polypeptides, strands, that wind around an imaginary axis, forming a double helix. Two sugar phosphate backbones run in opposite 5 to 3 directions from each other, anti-parallel, like a divided highway. Sugar phosphate backbones are on the outside of the cell, and the nitrogenous bases are paired in the interior of the cell. Two strands are held together by hydrogen bonds between the paired bases. This is important. Bold. Red. Adenine with thymine, guanine with cytosine. The two strands of the double helix are complementary, each the predictable counterpart of the other. DNA generates two identical copies of each DNA molecule in the cell that is preparing to divide. The structure of DNA accounts for its functioning of transmitting genetic information whenever a cell reproduces. RNA molecules exist as single strands. Complementary base pairing can occur between regions of two RNA molecules or even between two stretches. That looks better, yeah. It's two. I'm just gonna leave it, yeah. No, I don't know. Two stretches of nucleotides in the same RNA molecule. Complementary base pairing can occur between regions of two RNA molecules or even between two stretches of nucleotides in the same RNA molecule. Base pairing within an RNA molecule allows it to have 3D shape Base pairing within a RNA molecule allows it to have a 3D shape necessary for its function. Transfer RNA, or tRNA, brings amino acids to the ribosome during the synthesis of a polypeptide. In RNA, adenine pairs with uracil, and there is no thymine in RNA. DNA is always a double helix. RNA is more variable in shape. Okay. So that's it for concept 5.5. Concept 5.5, nucleic acids store, transmit, and help express hereditary information. Next video, we'll be going over 5.6, genomics and proteomics have transformed biological inquiry and applications. Okay, so I'll see you guys next time. Have a good one. Take care. Bye.